of Zoology, St. Joseph College, University Section, North Point, Darjeeling, West Bengal. As we have done so far during the course of last 10 days, we are organizing this kind of monsoon lecture series and of zoology, and I'm very grateful to Dr. Bera for giving us his precious time. And I also welcome Dr. Ritesh Vishwa, HOD, Gurugathan Government College, and the convener of this monsoon lecture series. And I request Dr. Vishwa to formally welcome Dr. Bireshwar Beda.
Thank you, sir.
Thank you, Dr. Sandeep Talukdar, my beloved colleague and junior. And we too have been doing this thing. Actually, the whole idea came from Sandeepan himself, and it's a beautiful work we have been doing for past 10 days. And it has been, I would say, it has been very much successful till now. I formally welcome Dr. Bireshwar Bera to this monsoon lecture series on behalf of Department of Zoology and from the family of Guruvathan Government College. Before I go ahead with formal invitation, I would like to give a brief introduction of Dr. Bera. Dr. Bera is an expert in fisheries, fish and fisheries. He is serving St. Joseph College for past many years. In fact, he joined St. Joseph College in 2010, right, sir?
Yes, sir. Yes, sir.
2010,
Yes, sir.
yes. And for past 14 years, he is there as associate professor. Before that, he served in different colleges at different capacity in the post of assistant professor, like in Netaji Mahavidyalaya, Ravindra Mahavidyalaya. He also was the project scientist and project fellow at the University of Calcutta and Institute of Environmental Studies and Wetland Management. He did his PhD in 2006, and he passed out his MSc in 1999 from the University of Burdwan. So many of you were not even born by then. So he
Yes,
is.
yes, yes, yes. Yes,
Uh, Dr. Bera is also the reviewer of many journals. Uh, as we can see that his profile speaks a lot. Uh, I mean, he has been the reviewer of African Journal of Agriculture, Asian Journal of Environment and Ecology, and so on and so forth. He has published a lot of publications, a lot of books, and he is an avid researcher. Despite being a teacher, he is also an avid researcher. And today, it's our honor to have Dr. Bera with us, and I hope his knowledge will definitely help us in knowing a lot about the Isoparochris, this trematode parasite.
yeah.
I welcome Dr. Bera formally to today's series, today's lecture, and I also welcome my students to today's lecture. I hope this session is going to be wonderful session. Welcome, sir. And
Thank you, sir. It is a great pleasure for me to attend this. And Ritesh sir is a very good person to me from the beginning of the from the Kalimpong College. We met together, I think so. From that time,
yes, sir, yes.
yes, yes. So thank you, sir, for your welcome address and for your assistance. So can I proceed, sir?
Absolutely, sir. Floor
Yes,
is yours.
yes. Thank you, sir. Thank you, sir. So today my topic is parasitic helminth trematode. That's one type of parasite which lived within the fishes. Actually, the specific fish is a celluloid fish. So I want to discuss about the carbohydrate metabolism. Which is, the study was not done before that. In reference of the parasite is the Isoparochus hypsilobacri. Uh, so it is prevalent in India and abroad also. And infection also occurs in humans who are consumed the raw fishes in the northeast Indian. So here you can find out what type of metabolism and what type of carbohydrates they are taking. So, you know what is the trematodes, students, for student purpose. Trematodes are actually the flatworms. You know that. Platyhelminthes. Actually, you know that the flatworms are fasciola, just look like the fasciola species. So it is a broad resembling the leaf or of the tree of the flatfish. So trematode species, there are many types of trematode species we have studied. Many types of researchers are published regarding these parasites: Schistosoma, Parampistoma TD, Echinostomas TD, or Pistoscori TD. But very rare and nothing, only in case of Isoparaches, which I studied in Calcutta University, and studying is going on still now. Uh, so I want to say about that the carbohydrate metabolism. So in most of the parasite, especially the helminth parasite, so their habitat actually the within the host or outside the host, ectoparasite or endoparasite, especially endoparasite, they absorb the host glucose actually for their energy purposes. So carbohydrate metabolism in nematodes, fascinating area, you know that. So parasitic flatworms, in that cases, are a unique process compared to their host. So host, there is a carbohydrate metabolism, actually the vertebrate hosts, and the parasite resides within that host. So their metabolism will be slightly different. So how they survive, either oxygen rich or without oxygen, how they adapted there. So you have to remember that the parasites like Schistosoma is reside within the host in the oxygen-rich habitat. That means blood parasite. Always oxygen supply is there. But some parasites which are not present in direct oxygen are muscle parasites. So how they survive there after the death of the host? So we know that glycogen storage of glycogen in case of Phaseola or any other species of helminths. So they stored the glycogen for their future purposes for the survive, I mean tissue. Okay. So trematode Phaseola species stored glycogen in their tissues. But when the food is not available, that means during starvation period. So I want to say about, remember that starvation period because it will be discussed later. Starvation period, they utilize this glycogen. Okay, how they utilize their glycogen and the how they break down their glycogen and which pathway they follow. This is our interest. How glucose is broken down in which pathway and glycogen in which pathway they utilize the metabolic pathway to survive within the host. And next, our interest is lactic acid production. Trematode metabolize glycogen is okay, but in products, after the metabolism of the carbohydrate, they produce a lactic acid. This is the significant because it indicates the anaerobic metabolism. You know that lactic acid is produced as the pathway where the oxygen deficiency is taking place. That means anaerobic metabolism, which is common in parasitic organisms. Most of the parasites, they produce the lactic acid as their end product of the carbohydrate metabolism. So source of energy is glycogen or glucose, and end product is lactic acid production. Not only lactic acid production, there is any other also end of products are there. We want to discuss about that and which pathway they follow. We have to find out. So questions will arise. Infection with trematodes. So there is the metabolic shift in their host. Okay. Because fermentation is taking place. Trematodes often rely on the fermentation process for energy production. Due to the low oxygen supply, it is the common things in case of parasite which are reside within the host. So this metabolic adaptation helps the trematodes to survive and thrive within their host and making them successful parasites for their habit. So, is there a specific aspect of carbohydrate metabolism in trematodes that you are particularly interested in? So, this is our question. Is there any specific aspect of carbohydrate metabolism in trematodes that you are particularly interested in? That is our question, and we are to finding about this. What type of metabolism is taking place there? So, trematodes, where the carbohydrate metabolism is adapted, they utilize the glucose, host glucose, as a glycogen. In other carbohydrates for energy. So how they obtain glucose from their host, either from blood who are present in the blood or who are present in the tissue from the intestinal fluids. And they store this glucose as a glycogen, as you know, I discussed from the beginning, which serves as the reserve of energy source. So you studied all things. The glucose metabolism is taking place through either EMP pathway, that means from glucose to lactic acid production or pyruvate production. So we know the steps. So pyruvate, in presence of oxygen, pyruvate enters into the TCA cycle. You know that. So through TCA cycle, they produce the ATP in presence of oxygen. But if there is no oxygen or insufficiency of oxygens, what will be taking place? So glycolysis, trematodes have functional glycolytic pathway similar to other organisms, and breakdown of glucose takes place into pyruvate. But in case of anaerobic respiration, that pyruvate is transferred into the lactate. But in case of oxygen-rich habitat, this pathway, that means trematodes, there is a debated issue. Either they directly enter into the TCA cycle or what is present there. For aerobic respiration, while others indicate the presence of functional mitochondrial electron transport chain is present or not. If functional mitochondrial electron transport is present, then pyruvate directly enters into the TCA cycle through the acetyl coenzyme, but there is no acetyl coenzyme. So how or what pathway they follow to operate this cycle? This is the question we have to answer. We have to find out which pathway they follow after pyruvate production to enter the oxygen, that means TCA cycle, not the directly to the TCA cycle. And anaerobic metabolism, that means capable of anaerobic metabolism means particularly in hypoxic condition. That means fermentative pathway to produce the lactate. So what type of adaptation? Enzyme regulation. So you know that in glycolysis, classical EMP pathway, some regulatory enzymes are there. If we if we quantify these enzymes, regulatory enzymes are, first step is the hexokinase from glucose. First step is the hexokinase, and from glycogen breakdown there is a glycogen phosphorylase. This is our most important enzyme. Pyruvate kinase. Pyruvate kinase is most important because that produce the pyruvate. And another enzyme which is not applicable in case of vertebrates is the most important thing is phosphoenol pyruvate carboxykinase, which is present in case of only mammalians parasite. phosphoenol pyruvate carboxykinase (PEPCK) are regulated to optimize the energy production, and carbohydrate utilization based on parasitic needs and environmental condition. So PEPCK. So you have to remember that pyruvate is not directly enters into the TCA cycle. So they follow the process through PEPCK to enter into the TCA cycle. So hexokinase isoenzyme, you know that hexokinase isoenzyme high affinity for glucose. Also in their body, that means capacity for glucose carbohydrate availability, where the glucose starvation free. What is the significance? Efficient carbohydrate metabolism is essential for nematodes for survivability, which enables them to thrive in the host environment and evade the host to start up the carbohydrate metabolism or any other metabolic pathway. So our targets to understand the carbohydrate metabolism, because develop antiparasitic drugs that targets key metabolic pathways. So, inhibiting parasite growth and survival. This is most important part. If we know all the catabolic or metabolic pathway which is operative within this parasitic system, then it is very much easiest to develop the anti-parasitic drug for that parasite. So, what is the challenges in this case? This exhibit variations in their carbohydrate metabolism, very much difficult. That makes the challenge to develop broadly the effective treatments, and less accessibility. Studying of metabolism parasites in vivo is very much complex and often limited by the availability of the suitable animal models. This is most important in case of research area. So overall carbohydrate metabolism in trematode is have the implications for understanding parasites biology and developing novel antiparasitic therapies. So this is our study. This is our model parasite, which is looks like fasciola hepatica. Middle is the fasciola. In the left side and right side is our target parasite, which is known as Isoparadis hypsilobacri, digenetic trematode. It is life cycle totally studied. Life cycle studied in the Kongsabodhi Reservoir. You know Kongsabodhi Reservoir is the Mukurmanipur, where all hosts are there. Is the Walawatu, okay, which is commonly known as boal. Boal fish. Adult is reside there, and their intermediate host is the Indoplanordis exustus and Mistas aur. Okay. So through these fishes, they complete their life cycle. So in case of nematodes, carbohydrate metabolism study has drawn less attention. So most of the study you can find in the whatever published cystodes and nematodes. Most of the studies are on the cystodes and nematodes, and fasciola cystosoma, all works are going on. So Isoparadis hypsilobacri is the parasite, fish parasite, which deteriorate the fish market value, okay, either mistress or wallower. So glucose and glycogen are the only energy source. Adult take up glucose and store as glycogen, but When you keep it in survival, in starvation, they deplete their glucose or glycogen. It was found that after receiving from the host, if you keep it in a culture in vivo studied, okay, in starvation period, they live for 15 to 20 days. How they survive and what is going on within this parasite. So it was found that they deplete the glucose and glycogen. So what's going on the metabolic pathway? So the level of free glucose in parasite is very much extremely low. Clear? So Helvis found oxygen poor habitat passes higher saccharide value when compared with that oxygen rich habitat. But Isopyrotis, this parasite, lives within the swim bladder of Wallawatti. Okay, Wallawattu is the oxygen rich habitat. Wallawattu is the oxygen rich habitat. In the living condition, it's okay. But when fish are coming in the market in the preserved condition after 15 days or after 1 month, parasites still alive within the swim bladder of the Walago R2 where there is no oxygen level. So there is three important aspects. We also know that the range of substrates of helminth can metabolize very much limited. That means carbohydrate, only energy source. Number two, acetyl coenzyme A, already discussed, does not occupy the same central role in case of metabolism. That means acetyl CoA directly not entering into the TCA cycle. Helminth as the mammals. So phosphoenol pyruvate appears to be the key metabolite. Phosphoenol pyruvate. Then finally, instead of degrading substrate completely to carbon dioxide and water, helminths characteristically produce complex end products, usually organic acids. This is our three important aspects of the study. So find it. So glycogen, glucose, glycolysis is taking place. Finally, the PEP, phosphoenol pyruvate. From phosphoenol pyruvate, what's going on? Pyruvate, pyruvate is going to the lactate. Clear? But pyruvate is not directly enters into the TCA cycle. Directly not enters into the TCA cycle that passes through the PEPCK. PEP PEPCK. So PEP to PEPCK oxaloacetate malate, that malate enters into the TCA cycle. That malate transfer into the malic enzyme to pyruvate, then acetyl coenzyme. The reverse TCA cycle you can find in case of parasite. So carbohydrate is breakdown through the EMP pathway similar to the vertebrate and at least as far as phosphoenol pyruvate. Then phosphoenol pyruvate is the main fat, which is the different from that vertebrates and helminths. This phosphoenol pyruvate is the main key factor or key substrate from where you can distinguish the difference distinguish from the vertebrates and helminths. The vertebrate TCA cycle appears, that means insignificant in production of energy in parasitic helminths, because here is the reverse TCA cycle is taking place, not directly. So phosphoenol pyruvate, find it, phosphoenol pyruvate through phosphoenol pyruvate carboxykinase to oxaloacetate to malate, that malate enters into the mitochondria. So this is the reverse TCA cycle is going on within the parasite. Is it clear? I think so it is clear. The pathway. So we have to now measure the what type of enzyme is working and how it is working during where there is no substrate, there is no glucose, glycogen during the survival. That means during the starvation condition. So two major pathways: phosphenol pyruvate, first carbon dioxide fixed by the PEP through the enzyme PEPCK, resulting in the formation of oxaloacetate, and then oxaloacetate is reduced to the malate by malate dehydrogenase. So malate then enters into the mitochondria. You already it was known from the diagram. So the reaction is then catalyzed by the mitochondria NAD-linked malic enzyme. So NADA is formed by this reaction is then used to reduce the malate to succinate through fumarate through the action of fumarate reductase. Now, so parasitic helminths produce what type of end products? Lactate, pyruvate, succinate, propionate, and frequently a major excretory products in helminths. They produce through the carbohydrate metabolism. So the outstanding feature of the carbohydrate metabolism in parasitic helminth is the production of reduced organic end products and this persists even under aerobic condition. So the range of end products produced by the helminth is, however, considerably less than that found in the microorganisms. So our model experiment, Isoparchus hypsilobacter, this is the digenetic trematode, reside within the swim bladder of the Walaguat2. This is the carnivorous fish, and metacarcaria found within the musculature and peritoneal cavity of the Mystacea. That is a non-carnivorous catfishes. So, habitat, that means swim bladder musculature, highly aerobic, but they are obligatory aerobic parasite because when oxygen is not available, they shift from aerobic to anaerobic. Ability to switch off over from aerobic to anaerobic is yet not known, but we want to finding out. Clear? So this is the Isoparachus, the digenetic trematode. It is a fish fluke. It creates the black spot disease or ink spot disease in a variety of fish hosts. Reported several Asian countries: Japan, China, Korea, Vietnam, Indonesia, Bangladesh, Pakistan, India, many countries. So our importance. So importance is most countries Walagat is a large number of other edible fishes. Okay, Walagat is market value is much more, like Mistas, Notopteras, Chana, okay, higher vertebrates like frog. So as such parasitic helminths are considered fishery sciences. So it has been known that members of the genus Isoparachus trematodes living in the air bladder of freshwater catfish in Asia and Australia. So find it. Walagat2, infected swim bladder filled with parasite. They live, they still alive after the death of the host. After the death of the host, they are still alive. So niche condition, oxygen rich habitat. Already you know that, and they survive without oxygen many days, 15 to 20 days. So distributed many countries, many fishes. So parasite utilize one particular freshwater pulmonate snail in the Planorbis exustus, already known that it is the intermediate host. So this is our study area, Kongsabati, Reservoir Mugur Manipur, where easily available all infected fishes, mistas, Walago, all are infected. This is the pathogenicity. Okay, swim bladder, liver also. So they absorb the glucose stored as the glycogen, then correlate to the energy expenditure, which again depends upon the activity of the worm, particularly the egg laying. Egg laying is most important part for survivability of the parasite. So our objective is to quantify the free glucose content and how this glucose is utilized during the starvation period. Okay, glucose is a primary source of energy, and how they utilize this glucose and glycogen during starvation period in vitro condition. And number three, metabolic pathway. Which metabolic pathway they follow to utilize this glucose and glycogen and produce the end product. So this is a method. I don't want to discuss this. So collection of parasite, already known that from the walawa to swim bladder. For starvation, the parasites was kept in the previous solution, simply previous solution without any glucose. Tissue extract for enzyme activity, all are done in the cold centrifuge and cold room. Okay, all chemicals, because very much highly thermostable enzyme. That's why all experiment was done in the cold room. So uh, our observation find it. Free glucose content very less, 0.6 milligram percentage. Glycogen storage is a fresh parasite. After collection from the host, it was measured that glycogen near about 5 milligram percentage. Hexokinase enzyme, most important enzyme for starting of the EMP pathway, 101.95. Glycogen phosphorylase, which is required for breakdown of the glycogen. Then most important enzyme is phosphoglucoisomerase. To study that phosphoglucoisomerase in the EMP pathway. Then pyruvate, then lactate dehydrogenase, because lactate is the end product. So LDH will help from pyruvate to lactate. And the PepsiK. PepsiK, you know that, for reverse TCA cycle, because directly pyruvate not entering into the TCA cycle. And TCA cycle enzymes is either TCA cycle is pursued within the parasite. To know about it, ICDase is the most important. ICDase, isocited dehydrogenase, alpha ketoglutarate dehydrogenase, and malate dehydrogenase. These are the most important enzyme. And end products are found that phosphenol pyruvate, lactate, oxaloacetate, malate, and many more are there. So these are the fresh parasites after collection from the feces. So then we kept the fish parasites within the previous solution in various hours, 6 hours, 12 hours, 18 hours, 24 hours, to up to 72 hours. We experimented that and found that from the graphical representation, it was continuously decreases. Glucose content is continuously decreases. That means glucose is utilized during starvation for their survivability. Glucose is utilized during starvation for their survivability to after 72 hours, 0.15, from 776 to 0.15 milligram percentage. Glycogen content, find it. Glycogen also content is decreasing. The rate of decreasing is much more after 24 hours. Up to 24 hours, it's okay. So 5.127 to 1.6 milligram percentage. after 72 hours. So we are finding out that glucose and glycogen both are decreasing during starvation. That means enzymes also working on that system. How enzymes are worked there for breakdown of glucose and glycogen. So first enzyme. So this is the leakage of glucose. We have to estimate that because through the cell membrane of the parasite, through the parasite in the culture media, there is previous solution. Glucose transporter through glucose transporter, some glucose is released out from their body. So we have to also estimate that the is there any leakage of the glucose from the parasite or not for confirmation. The either metabolic pathway that is glucose utilized or is there any leakage of glucose taking place from the parasite body. So we are finding out that very minimal quantity, microgram, okay, only microgram percentage parasite is a leakage of glucose is taking place from their body. It is very minimal, so it is not in our consideration. Now, rate of uptake, that is after starvation period, if you put some glucose in the culture media, you can find that they also take the glucose from the culture media. Okay. So from the beginning, they are not able to take much more glucose. But time slot when you increasing that after 12 hours, the rate of uptake is increasing. So control fluke, that means control fluke means after releasing from the host, and experimental means after starvation period, if you put the fluke into the glucose media, what percentage of glucose get released per hour? So rate is more or less similar in case of either starvation or after taking the of uptake of glucose from the other source. That is the rate. Find it more or less same. In the beginning very much high, then it will be saturated. Various concentration is there. Okay. Now I want to say about that our main things is enzymatic analysis. Hexokinase enzyme, the first enzyme for breakdown of glucose. What is the fate of the hexokinase enzyme? So hexokinase enzyme in control fluke means that after collection from the host, that is 101 point, that means 102 near about nanomole per minute per milligram protein. So it was starved for 72 hours in without glucose. So 6 hours, find it is decreasing. Hexokinase activity is decreasing at 24 hours. Up to 24 hours, the decreasing rate is much more. Then it is slightly normalized. Decreasing rate is going on, but after 50 hours, the decreasing rate is much more to 72 hours. That means from 101 to 20. That means hexokinase is utilized or hexokinase is performed within the parasite for breakdown of glucose. So glucose to glucose 6 phosphate. Glucose to glucose 6 phosphate. That means EMP pathway is going on in the primary level. But glycogen also decrease. We found there from the starvation period, glycogen also decreasing. So glycogen is two types, glycogen A and glycogen B, glycogen phosphorylase A and B. Okay, glycogen phosphorylase A is responsible for breakdown of glycogen, not the glycogen phosphorylase B activity. Glycogen phosphorylase B activity actually active in case of vertebrate species. So we are take in account the glycogen phosphorylase A activity. It was found that glycogen phosphorylase A activity is increasing because glycogen phosphorylase A activity is increasing. That means glycogen is breaking down. Glycogen is breaking down. That means from 5.24 to 77.53 specific activity, it was found that increasing glycogen phosphorylase A activity is increasing. That means glycogen is breaking down. If it is reduced, that if the concentration of the glycogen phosphorylase A reduced, that means there is no breakdown of the glycogen. When the glycogen phosphorylase A is active, that means breakdown of glycogen is taking place. So glycogen is breaking down from glycogen to glucose 6 phosphate, glucose 1 phosphate and glucose 6 phosphates. Glycogen to, so breaking down is taking place. Find it. Then our next target enzyme, phosphoglucose isomerase. Phosphoglucose isomerase PGI activity. It was found that same tradition is going on, but not much more of hexokinase. Not much more of hexokinase, but hexokinase, in case of hexokinase, breakdown is much more of the glucose. But phosphoglucose isomerase, it is gradually decreasing, not look like of the hexokinase. But trend is decreasing of the concentration of the enzyme. Concentration of enzyme is decreasing in the starvation period from 6 hours to 72 hours. Same things, pyruvate kinase PK also is taking place. Now, lactate dehydrogenase, that means LDH activity, that means pyruvate kinase activity is the last step is the lactate dehydrogenase. That means lactate production is going on in case of anaerobic condition. That means parasites are placed in the in vitro starvation condition. Parasites were take place in the in vitro starvation condition where oxygen supply was not there. That's why breakdown of pyruvate is taking place very much fast from 104 to 21 nanomole. So very much clear that when parasite lives within the host swim bladder, where is the oxygen is a habitat, where the lactate is 104, where lactate production is 104. In case of oxygen decreasing condition, that means in the culture media where oxygen supply is not available, that means they have the capacity from oxygen rich habitat to low oxygen rich area. So transformation of oxygen rich habitat to, that means aerobic condition to anaerobic condition is taking place from this lactate hydrogenase enzyme activity. It is clear? I think it is clear. Now, the main target that find it lactate dehydrogenase activity. This is the PEP-CK. That is the turning point of the parasite. Pyruvate is not entering into the mitochondria directly, so they bypass to the phosphoenol pyruvate carboxykinase enzyme from phosphoenol pyruvate. So it was found that if PEP-CK is increasing during the starvation time period. That means from phosphoenol pyruvate, the parasite, for their survivability, they follow that process. That means phosphoenol pyruvate carboxykinase, that is increasing trend from 172 to 1545 nanomole, increasing taking place during that starvation time period. Then the breakdown is going on through the PEPCK from phosphoenol pyruvate to malate formation. Now, if there is TCA cycle is operative, so isocited dehydrogenase is the first enzyme which determine TCA cycle is taking place. So ICDH NADP dependent, where you can find that NADP, ICDH NADP dependent, that means ICDH in the control condition, in the phase condition, and in the stop condition, it is decreasing. That means breakdown. That means parasite utilizing the TCA cycle. That means minimal oxygen they are utilizing through their mitochondrial part. Alpha ketoglutarate dehydrogenase. This is also the TCA cycle enzyme. That means TCA cycle is operative, and malate dehydrogenase activity, which is also the part of the TCA cycle. That means parasite enters through the PepsiK to malate. Malate enters into the mitochondria. Within the mitochondria, then the malate transform into the acetyl coenzyme A that passed through the ICDH and TCA cycle. Cycle is of TCA cycle. Both are operative in case of parasite. So this is a phosphoenol pyruvate with a major substrate as well as major end product in case of parasite. So from phosphoenol pyruvate through PepsiK to the TCA cycle. So PEP is decreasing trend. PEP showing the decreasing trend, and PepsiK is showing the increasing trend. So for breakdown of the PEP, PepsiK is responsible for that. For breakdown of PEP, PepsiK content is increasing. As PEP is decreasing, phosphoenol pyruvate carboxykinase is increasing. So then pyruvate, that means from PEP, pyruvate is also produced, which is also showing that find it increasing trend. That means end product, end product of the carbohydrate metabolism, pyruvate content, already known that that is increasing. Then lactate content is also increasing as an end product of the carbohydrate metabolism. So PEP increasing, pyruvate increasing, lactate content increasing. Then oxaloacetate content increasing, which is the end product of the TCA cycle or substrate, whatever is there. Oxaloacetate content is increasing. Then malate content is increasing. Find it. Malate content is also increasing. All end products are increasing due to the breakdown of carbohydrate. So the pyruvate kinase and phosphoenol pyruvate carboxykinase ratio, which determine carbon dioxide fixation is going on or not. This ratio say that there is carbon dioxide fixation, that means not proper TCA cycle,